Hello cute little champions, I welcome you all to mock test 9 of our NEET Premier League, and today in this particular mock test I have included most amount of questions from evolution. Then comes a new chapter that we are dealing of as of now, that is biotechnology principles and processes. But as I said, most of the questions will be from evolution, a little bit from this chapter and a little bit from the chapters that we have already completed. For example, sexual reproduction, flowering plants, or probably principles of inheritance and variation. You can guess. Okay, but anyways, you would be finding 1 or 2 questions from here and there. Chalo, let's begin. Okay, according to Darwin, the organic evolution is due to, who was Darwin? Basically, Darwin was a big man who had his own theory of evolution. He said that basically there is something that is known as natural selection. What does natural selection mean? That means if an organism has good amount of variation and this particular variations is making it different from other organisms and is making it survive during unfavorable condition, then this organism will be surviving. So survival of the fittest and everything was given by Darwin. Okay. So basically, according to what did he give the organic evolution? So according to Darwin, the organic evolution is due to, why there is evolution? Basically, competition within closely related species, is that the reason why there is evolution? Or because reduced feeding efficiency in one species due to the presence of interfering species, or due to intraspecific competition? What do you mean by intraspecific competition? That is competition in between, among the species itself. For example, this is a group of species A. Now there is a mango. Okay, there is very limited amount of mango, but all of them want mango. Over here, they all will be fighting amongst each other for the food, okay, for the food or the resource basically, and this type of competition is known as intra-specific competition. Now you only think, if internally within a species only competition is having, they do not have a choice rather than to evolve. So obviously they will be evolving. They will undergo evolution, and this was the reason that Darwin gave. According to Darwin, the organic evolution is due to intraspecific competition. Intraspecific competition as in, there is another species that you see over here, and that is B. Is it because of intraspecific? Sorry, interspecific, the competition between them and them, that a new species arose? No, children. It's because of intraspecific, the competition within or among one particular species, that led to organic evolution. This came in NEET 2013. Going ahead with the next question. Dinosaurs dominated the world in which of the following geological eras? So dinosaur, I can say they came in one particular era, that is Mesozoic era. They came in one particular era, that is Mesozoic era. And if you're thinking, oh ma'am, this is given, so this is the correct answer. No, children. The question is, dinosaurs dominated the world in which era? So that is something else. So Mesozoic era, I can divide it again into Triassic period, and then Jurassic period, and then Cretaceous period. So over here I say this is the time where the dinosaurs actually came to be of use. They appeared. Okay. So dinosaurs came during this particular point of time. They kind of dominated the earth during Jurassic period, and then in the Cretaceous period they were about to get extinct. So this was Mesozoic era in which all of this happened. But what was the phase where the dinosaurs were dominating, children? That is the question. So it would be Jurassic phase. Okay, so this is the era during which they were dominating, and this is the question from Karnataka NEET 2013 yet again. Going ahead, we have question number 3. According to the fossils discovered up to present time, origin and evolution of man started from? According to the fossils discovered up to the present time, origin and evolution of man started from what time? I read the question in a different form. Okay, where do you think that human beings, or basically man, evolved? You have options as France, Java, Africa and China. Now Java comes in Philippines. Okay, Java comes in Philippines. I might have messed up with the spelling, but I know you know the spelling. Anyways, one thing before that, so I just started off with a mock test, but I want to tell you, you should be seeing this video only if you have tried it by yourself. So you have the question paper given to you in the community post, okay, or in the WhatsApp group. You have to try it out, my dear children. Without trying it out and just relying on me won't do any good. Okay, I do not want your views. I want you to practice. Now, if you have practiced, then come see and check whether your answers are correct. If your answers are correct, give yourself +4 marks. Okay, or if your answers are not correct, give yourself -1 mark and then only go ahead. This is an analyzation of your preparation. Okay, so here there is no cheating involved. Here you have to try it out and find out. Okay, so evolution of man, France, Java, Africa, China. I'll say Africa. It was in East Africa, where you saw Dryopithecus and Ramapithecus. Basically all of them, it is Ramapithecus through which we evolved into a man. So basically it was East Africa where everything happened or is believed to have happened. Okay, so this becomes the correct answer. This came in NEET 2002. Now, which of the following had the smallest brain capacity? Okay, what are these organisms that you see over here? Basically, this is the evolution of man. So, among this, if I say, the first guy that have appeared is Homo habilis. Okay, then from them, Homo erectus have evolved. Okay, then from them, I'll say two things. Homo neanderthalensis and Homo sapiens. So it is believed that it is divided into two, but yeah, they came first and they came second later on. Okay, but anyways, it went off like that. And you know, in evolution of man, obviously the brain capacity has to increase because we human beings have enough brain capacity, even though we do not use it much, but we definitely have. So the brain capacity chart, if it goes like this, I'll say brain capacity is measured in terms of cranial capacity. Okay, so over here Homo habilis is having 650 to 800 cc cranial capacity. Okay, now Homo erectus is having 900 cc brain capacity. Okay, Neanderthalensis has 4,400 cc. It is said, not given in your book, but it is said Homo sapiens had 1350. Okay, 1350 cc. Fine. So if you see, they have evolved to something better. Now if you are confused with that regarding this, okay, the number is not given in your NCERT, it can be much higher, but according to some other textbooks and all, it is 1350 itself. But anyways, if you see which of the following had the smallest brain capacity, that is being asked. Okay, what is it in an NCERT? That is only being asked over here. So as you can see over here, it is Homo habilis which has the lowest brain capacity. So can I put a tick mark on it? Yes, I can. This came in NEET 2015, my dear little champions. Okay, going ahead. Fifth question, which of the following statement is correct? Okay, Australopithecus is the real ancestor of modern man. Chalo. So we'll go back to this question only. Okay, and I'll say before that you had Australopithecus. Before that you had Ramapithecus. Okay, along with it you had Dryopithecus. Okay, they were more like ape-like and these guys were more man-like. So this is how everything has evolved. Okay, now instead of Homo sapiens, it is always written Cro-Magnon-Man. So please understand, it's the Cro-Magnon-Man which has given rise to Homo sapiens. So there might be a lot of steps which is not included in your NCERT, but it's actually there. So you should be knowing it. So Cro-Magnon-Man, if we see that, just understand we Homo sapiens have come from them. Okay, Neanderthalensis, they are different. They are different. Cool. Now, yeah. Which one of the following statement is correct? Australopithecus is the real ancestor of modern man. No, children. Who is modern man over here? This guy, Homo sapien, is the modern man. So do you think that Australopithecus is the ancestor of modern man? No, children. You need to find the commonest, the nearest ancestor. Okay, so it is somewhat true, but not exactly. Neanderthal man is the direct ancestor of Homo sapiens. Again, Neanderthal man is the direct ancestor of Homo sapiens. No, it's not the direct ancestor. Going ahead again. Are you? Yeah. Homo erectus is the ancestor of man. Chalo dekhte hain. Homo erectus is the ancestor of man. Kind of true. Yeah, but there is one more option left. Cro-Magnon man's fossil has been found in Ethiopia. No, children, it's not found in Ethiopia. That's wrong. So we'll say Homo in Ethiopia and Tanzania. No, you found Australopithecus before that. Okay, so that was something entirely different. You don't have to go back until that. It's the Homo erectus is the ancestor of man. This becomes the correct answer. Okay, and how does ma'am remember all this thing? Basically, if you revise it often, you will be remembering. The first time I saw it, no, I'm truly saying, I was like scared to learn all these things. Okay, but I was like, no, I have to anyhow learn it. There is no option. Don't think of it as an option, you will study it. As simple as that. Chalo. Next, question number 6. Which of the following statements is correct regarding evolution of mankind? Homo erectus, okay, is preceded by Homo habilis. Okay, proceeded and preceded is different. Okay, preceded as in it is before. It is before. Okay, Homo erectus is preceded by Homo habilis. As in this particular thing is before this particular thing. That is what the question says. Okay, rather than making you more confused, I'll just rather explain it to you that this particular statement says that this guy has come before this guy, has preceded before this guy, has come before. Okay, you can remember like that for preceded term. Okay, preceded as in basically after this. Okay, the next thing that will come is proceeded. This is preceded. Fine. So, which of the following statement is correct regarding evolution of mankind? Homo erectus is preceded by Homo habilis. So, before that, erectus, did you have Homo habilis? Let's go and check over there again. Yes, before Homo erectus, we had Homo habilis. That is correct. But we had to see other options as well. Neanderthal man and Cro-Magnon man were living in the same time, no children. Neanderthal man that you see over here, they came around 1 lakh years ago to 40,000 years ago. To 40,000 years ago. Okay, talking about these guys, they came around 75,000 years to 10,000 years ago. Okay, so they shared a common time somewhere, but it was not like they arose at the same time. No, that is entirely wrong. Okay, now, yes, Australopithecus was living in Australia. No children, they were all living in Africa. Don't go with the name. Now, none of this is obviously wrong, so this becomes the correct answer. 1997 question. Now, which of the following is the direct ancestor of Homo sapiens? Now you tell me. Homo sapiens direct ancestor will be Australopithecus? No. Homo sapiens Neanderthals? No. Homo sapiens fossilis? No. Homo erectus direct ancestor. Easy question again. 1996. You see the number of questions that have come from this topic, which is why I said it's important. Now, which of the following is correct order of evolutionary history of man? You're like, ma'am, you didn't teach this. Where is this? Where has this come from? Basically, if I say Homo erectus, no, Homo erectus, they are also known as Java man. They are also known as pecking man. So don't get confused. Why? Because until now everything was happening in Africa, but some Homo erectus species, I'm not saying everyone went off. So they were like, haha, we are done with chilling in Africa, we will go to China, Philippines and things like that. So these guys went to Philippines. That's why the Java man name come, because there is a place like Java in Philippines. Okay. Now talking about pecking man, again this is something related to China. Okay. So it's Homo erectus only, you're talking about them only. Okay. Now, after Homo erectus, what do you have, children? You have Neanderthals and you have the Cro-Magnon man, or basically the Homo sapiens. So I told you, in between this, in between the modern man, that is Homo sapiens and Homo Neanderthals, there could be something else as well, and that something is Heidelberg. That something is Heidelberg, which is not given in your NCERT, but you should be knowing it. So after Homo erectus, we had Heidelberg. Heidelberg later divided into this and this. Okay. So pecking man, Homo sapiens, Neanderthals, Cro-Magnon man, no children. Okay. Homo sapiens, as in basically it should be coming after Cro-Magnon man, because, okay, so this, then Cro-Magnon man over here, Cro-Magnon man, and then Homo sapiens. Okay. But in your NCERT they didn't mention about this and this, but you should be knowing. It was Heidelberg which divided into two, and now you had Neanderthals and Cro-Magnon man. Cro-Magnon man later came down to give what? Homo sapiens, the modern man. So pecking man is there, yes, pecking man is there. Then Heidelberg man is there, yes, then Heidelberg man is there. Then Neanderthal is there, then Cro-Magnon man is there. Okay. Now, if this is not clear, I don't want to confuse you, I'll write it properly. I know I sometimes mess it up, but I can help you with that. Okay. Okay. Cro-Magnon-Man. Cool. It goes like that. So this becomes the correct answer. 2001 question. Okay. Now going ahead. Which of the following statement is correct about the origin and evolution of men? Again, agriculture came around 50,000 years back. No children, it came around 10,000 years back. These are all important things. The Dryopithecus and the Ramapithecus primates existing 15 million years ago worked like men. So the first guys I told you that came was Dryopithecus and Ramapithecus, right? Dryopithecus has ape-like feature, Ramapithecus had man-like features. So both of them, they definitely came around 15 million years ago, that is true, but they didn't walk like man. They did not walk like them, they walk like gorillas, they walk like chimpanzees, okay? So this becomes wrong. Now Homo habilis probably ate meat. Now Homo habilis, as I said, after Australopithecus, you had someone, the first true hominid, human-like organism was the Homo habilis, okay? It was the first guy to have the term Homo in front. So Homo habilis definitely were human, very much human-like, and they were not the people who were liking to eat meat and things like that. Initially, they were the people who were eating fruits, my dear children. They were eating fruits, but they were excellent tool makers. So they were excellent tool makers. From them came the Homo erectus, which were feeding on meat. Okay, so Homo habilis probably ate meat. This is also wrong. They ate fruits. They ate fruits. Now Neanderthal men lived in Asia between 1 lakh and 40,000 years back. I told you, yes, they lived during this particular point of time. So this becomes correct. Okay, just give me a second. I'm feeling so hot. Okay, cool, works. Now 10th question. A question from evolution again. Genetic drift operates only in, what do you mean by genetic drift? Genetic drift means sudden change in the gene frequency or sudden change in the allelic frequency. For example, over here, okay, I made this example so many times, I know that, but I don't have any option. So there were green moths, okay, there were green moths and there were yellow moths. The green moths kind of dominated the area and said like, huh, you guys have to stay away from my place, this is our place. But then one day, what happened? One big elephant came and stamped on this, okay. One big elephant came and stamped over here, such that all of them, they got killed. So you had an allelic frequency, you had the gene frequency of this and this over here. So X and Y both were there over here, okay. But over here, now when the elephant came and stamped, you just had them. Now there is just a frequency of Y. You see the shift over here. There is a shift in the gene frequency and this is occurring by chance. This is occurring by chance, my dear population. My dear population, oh my god. I hope no one makes a meme on it. Okay, so my dear children, not my dear population, my dear people. Anyways, so this gene frequency change is happening and this is occurring very by chance itself. It's a random process and obviously it will show its effect only if the population size is small. Only if the population size is small. Now imagine that there are so many green moths over there, your big population size. So just one stamp by an elephant, will it cause much change over here? No. Only if the population size is smaller, that's when it will show its effect, which is why I am saying smaller populations. Okay, this is 10th question. Going ahead with the 11th question. In the developmental history of mammalian heart, mammals like us, okay, it is observed that it passes through 2-chambered fish-like heart, 3-chambered frog-like heart and finally 4-chambered stage. To which hypothesis can this be cited, this above cited statement be approximated? Oh well, listen to me very carefully, okay. So I say during embryogenesis, we had 2-chambered heart, and then 3-chambered heart, and then finally 4 two-chambered heart. Okay, now you should be knowing that two-chambered heart is found just in fishes. This is found just in amphibians and reptiles, and this is found in mammals and aves, that is birds. So basically I am trying to say that we had this type of heart, this type of heart and this type of heart. That means probably we might have evolved from these organisms, which is why we are showing their character as well. Now this was said by Ernst Haeckel, my dear children. This was said by Ernst Haeckel, according to which he put down a law which states, which is known as Biogenetic law, and it talks about ontogeny repeats phylogeny. What do you mean by ontogeny? Ontogeny means embryonic development. Ontogeny means embryonic development. Phylogeny means evolutionary history. Ontogeny repeats phylogeny. So basically during the embryonic development you are repeating the evolutionary history. That means we have evolved from them, that is being repeated, and this is said by the biogenetic law, my dear children. Okay, similarly one more example is there. Basically during the embryonic stage of us mammals, basically you could see this thing, what do you call it, gill slits. Do we have gills? Have anyone of you seen your fellow human being to have gills? Basically no one of us has the gills, but it shows that probably fishes had gills and we might have evolved from them, which is why the embryonic development, in our embryonic development also we also have the gills. Got it? So that is one thing. Now next question. Variation caused by mutation as proposed by Hugo de Vries are what? So there was Darwin initially who said that, okay, so in a group of organism there is some variations occurring. So somehow I do not know what is the cause of variation, but some variations are really good and these good variations are letting organisms live. Okay, some good variations are letting organisms live, and this is a very small variation. It takes long time to show its effect. It takes billions of years to show this effect and evolutionize into something else. Then came a guy who followed Mendelian, okay, who was very much aware about the genetics, and he said the variations that you're talking about, it arises because of certain mechanism which is known as mutation. So mutation means any changes in the DNA or the genetic material, that is mutation. So this is the reason why there are variations found in different organisms. Now this mutations that you see over here, it's a very random process. It's a very random process. It can happen anytime, anywhere, in whatever part of our genetic material. Okay, it can happen anytime, anywhere, in whatever part of our genetic material. So this is a very random process, and it is not having any direction. That means it will not obviously always result in something positive. It can also result in something negative. For example, you get diseases because of mutation, you get cancer because of mutation. Now this does not have a direction, does not say that I will always go to the positive side. No, it is directionless. So mutation is, children, it's a random process and it is directionless process as well. So this was said by Hugo de Vries. Okay, and this question came in NEET 2019. Works? Chalo. Next question. 13. Hummingbirds and hawk illustrate. So hummingbird, hummingbird is a bird, and then you have hawk, my children. Okay, what are they illustrating? What are they trying to show up? Basically both are birds, so I cannot say that they are showing homology, because homology means, homology comes when you have two different type of organisms but they have the same evolutionary history. For example, I talk about cheetah and man. Okay, so man is there, man is a mammal, cheetah is also a mammal, but two different species and everything is there. Okay, now one has four limb for having food, other has four limb for running. Over here, if you talk about the structure of the forelimbs in the case of cheetah and man, it's the same. The structure is same, but the usage is totally different. The function, if you see, is totally different. Over here, hummingbird is also a bird, hawk is also a bird, everything will be same in them. So I cannot talk about homology. Okay, I cannot talk about homology. What then? What else? Convergent evolution. What do you mean by convergent evolution? Convergent evolution means two different organisms are coming towards the same point because of the environmental things. For example, I have a butterfly and I have a bird. Both are of different things. These are insects, these are birds. They both are flying because their habitat is requiring them to do so. Get it? This is known as convergent evolution, where they have certain structures which are functionally same. Not anatomically, structurally they are not same. Functionally, it's just both of them have wings and both of them use it for flying. But anatomically, if you say the wing of the butterfly and the wing of the bird will be totally different. That is convergent evolution. Okay, that is the opposite of this. Now, adaptive radiation. What do you mean by that? Adaptive radiation means you have a common ancestor. You have a common ancestor, and from this ancestor, you are giving rise to other organisms as well. So, hummingbird, hawk, they are birds still, children. They are birds. Now, because of certain advancement in them, certain changes in them, okay, some variations in them, they went to different areas, geographical areas, and they got adapted there. Okay, they had the features to get adapted there. So, this thing is shown by hummingbird and hawk. Both of them are different things, but might have had a common ancestor from which they have evolved into different things. But still they are birds. But still they are birds itself, children. So, this becomes the correct answer. This came in 1988. So old question. Yeah. Next question. Question number 14. Study of fossils. What do you call study of fossils as? Study of fossils is known as paleontology, my dear children. If there is any Friends series fan over here, they might get it. Ross, the character Ross, he's a paleontologist. So he's used to work with the skeleton and things like that, the fossils and everything. Okay, so this is paleontology, and this is what you mean by study of fossils. Okay, next question. Which of the following is a pair of homologous organ? I told you homologous organ, whenever the term comes, homologous organs, just understand that functions have to be different. Functions have to be different. What about the structure? Structure would be similar. They will have similar structure anatomically, more or less. Okay, more or less, not entirely similar. Okay, so why did this happen? Basically, again, their environment forced them to have different functions. Cheetah cannot walk like this because their environment says so. Okay, we cannot walk like cheetah forever because our environment says so. We can just walk around freely. So the functions are different, but again, the forelimb structure and everything is same. So whenever you see the anatomical structure, they are evolutionary related, children. These guys, whoever will be having homologous organs, they will be evolutionarily related. Okay, and this type of evolution, which gives rise to different organisms from the same common ancestor, you call this as divergent evolution. You call this as divergent evolution. Okay, so now, whenever you find the same function, just exclude them. Okay, that is not the correct answer. So, wings of grasshopper and wings of crow, no. Lungs of rabbit and gills of rohu, lungs of rabbit and gills of rohu, they do not have the same structure. Even though they are used for breathing, again, this is the same function, but they are used for breathing anyways. It's not the answer. Wings of bat and wings of butterfly, again, the same thing. Okay, same structure, same function. Not exactly same structure, but yeah, same function. Over here also, same function is there. It's for respiring, breathing. But pectoral fin of rohu and forelimb of horse. Over here, yes. So instead of having forelimb in the case of fishes, they have pectoral fins. Okay, and that is, so this and this might have had some common thing earlier, but yes, their functions, if you see, they are totally different as of now. So this becomes your correct answer of homologous organs. So these questions are kind of tricky, but you will get to know about it if you practice it often. Okay, it's easy. It's easy. Now going ahead with the 16th question. The presence of gill slits in the embryos of all vertebrates supports the theory of what? Metamorphosis, biogenesis, organic evolution and recapitulation. So basically over here you might get confused with biogenesis because I spoke about biogenetic law. I spoke about biogenetic law which states that ontogeny repeats phylogeny, which says that ontogeny basically what? Ontogeny means what? Embryonic development is going to repeat phylogeny. Phylogeny means what? Basically it is going to repeat the what? Evolutionary history. Got it? Works? Chalo. So can this be the answer? Biogenesis is not the answer. Recapitulation is the answer. So ontogeny repeats phylogeny or recapitulates. Recapitulates phylogeny. So this becomes the answer. Biogenesis is something different. Biogenetic law is something different. So this is the answer. Please do not get confused. This came in 1995, my dear children. Okay. Now going ahead, 17th question. Variation in gene frequency within population can occur by chance rather than by natural selection. This is referred to as, I told you, whenever the gene frequencies change, and that is true because of chance. It's a very random process. Just to understand, it is genetic drift. It is what, children? Genetic drift. Okay. So random mating and things like that, no, it's not. Random mating also is not exactly random. Okay, we cannot say it is occurring by chance. People are deciding and then they are mating. Organisms are deciding and then they are mating. Yeah. And genetic flow means basically it is not occurring again by chance. Genetic flow means, for example, I have a population over here in Australia and I have a population of humans over here in India. From here, the white people are moving towards India. So that means their genes also, their body has genes, okay, they are also moving along. So I am carrying this gene there. This is known as gene flow. So here that particular creates a void, okay. Here that will change the gene frequency over here. When it moves there, it will add up something there. So it will change the gene frequency over there as well. This is known as gene flow. This process is known as gene flow. This is not a random process that is occurring by chance. When you talk about genetic drift, it is a random process which is occurring by chance. So this becomes the correct answer. Cool. That is the 17th question. Going ahead, we have the 18th question. Basic principles of embryonic development were pronounced by. Okay, I told you already the embryonic development. Who was the person who gave down the thing? It was Haeckel. What was his actual name? Ernst Haeckel, my dear children. And one more very important thing. There was one more guy, okay, in the troop. In the troop, his name was again Ernst. But it was born where Ernst, something like that. This guy, he said that whatever Ernst Haeckel is saying, it is wrong. It is wrong. And what did he say? Ontogeny repeats phylogeny or recapitulates phylogeny. He said that this is not true. In that case, it's not like, okay, we have the embryo in which fish's gills are present, but amphibian feature is not present. Reptilian feature is not present. So how can that be true? If we have evolved from fishes, then fishes evolved into amphibians and then amphibians into reptiles, then we should having the features of other organisms as well. Why is it not there? So I cannot say that ontogeny, embryonic development, repeats evolutionary history. That won't be really correct. This was said by this guy. This is given in your NCET textbook. Please read through it. So that question is also asked. Okay. So over here, the principles of embryonic development was given by Ernst Haeckel. No doubt regarding that. This becomes the correct answer. But there was one more Ernst who said this was wrong. Okay. And the question has also come. I think I have discussed when I was talking about this in the normal videos. Okay. Conceptual videos. Now, the phenomenon of industrial melanism demonstrates what, children? The phenomenon of industrial melanism demonstrates what? Industrial melanism, if you remember, you had two plants. Okay, this is before all the pollution and everything happened. It is pre-industrialization era. Pre-industrialization era. At this point of time, what happened was, It was pre-industrialization, there was no much pollution and things like that. So there were lichens all over. Okay, what did you have, children? Lichens. And what is lichens? Lichen is a symbiotic association of algae and fungi. So it is found only at the places where pollution is not that much. Okay, now post industrialization, the lichen won't be found because it is very much, it does not like pollution at all. It is a pollution indicator. It won't be growing where pollution is there. So you didn't have lichen over there. So this plant which is there, the bark of it was white in color, whereas over here because of dust and everything it was dark in color. Now there were two different moths, children. Bist and Betullaria. Okay, there were two different moths. One was white in color. This is the white one. And one was black in color. Okay, so what happened was, the white moth said, I am the one who rules the world, and it stayed like that over here, and no one was able to spot it because everything over here was white in color. But the black moth, whenever it came and sat over this, easily it was visible by the predators. Okay, so predators were like, the black thing is visible, it is a contrasting thing, so I'll go eat the black one. So nature selected the white one initially, but later on when industrialization and everything happened, the white lichen and everything went off. So you had a black background. In black background, if something white comes and sit, this will be visible. So now predators were like, haha, new taste, I'll just eat this. So this is a new thing that came. But the black ones, they were able to properly hide over here. Now if I make a black moth over here, even you won't be able to see it. Are you able to see it? No, because it is camouflaging. But the white ones, they were not able to camouflage, and this time the nature selected them. So this is known as industrial melanism. Melanin is a thing that is responsible for skin pigmentation, the coloration of the skin. Okay, so this is what you mean by industrial melanism, and this demonstrates, children, natural selection. The nature itself is selecting the best one. Survival of the fittest. Okay, so basically this was given by Darwin. You have to remember it. Works? Chalo. Next question. The similarity of bone structure in the forelimbs of any vertebrates is an example of. So similarity in the bone structure of any vertebrates, as I said, for example cheetah, for example us, human beings, or any vertebrate as in a fish or us, whatever you take, okay. If the structure is similar, my dear children, I told you, if the structure is similar, and if they have different functions, then this has to be homologous. Homologous, as in the same structure, they have structural similarity, but they have different functions because they have evolutionarized in different ways because of different adaptations that was needed by them. So that is the thing. Okay, so the correct answer becomes what? The similarity of bone structures in the forelimbs of any vertebrate is an example of homology. Got it? This came in NEET 2018. Okay, whenever you find similarity of the structures, internal structures, it is homologous. Whenever you find differences in the structures, anatomical structures, that means you're talking about anatomical hormone, analogous organs. Okay, so analogous is different, and homologous is this. Here the structure is similar but functions are different. Over here it's kind of opposite. Okay, structure is different, functions are same. Got it? Done? Next thing, 21. Choose the correct pair from the following. So this is the question from the chapter that we are dealing as of now, that is biotechnology principles and processes. So are you ready? Chalo, let's solve it. First question, we have to find the correct pair of enzymes. Okay, so ligase joins the two DNA molecules. Is it true? Do you think that ligase is that thing that joins the two DNA molecules? Let's see. Polymerases breaks the DNA into fragments. That is entirely false, because if we have DNA polymerases or any polymerase, for example A, T, G, C is there in this strand, DNA polymerase will form in 5' to 3' so this hash. So it will form the complementary strand A, T, C, G. This is the function of DNA polymerase. It is helping in replication. RNA polymerase helping in the formation of RNA, whatever. Okay, but anyways, it is helping in a formation of new strand. Does not help in breakdown. Nucleases. Nucleases means the enzymes that breaks down. Okay, cuts the DNA. Okay, not breaks down the DNA. It's basically the one that cuts the DNA off. That is what you mean by nucleases, or DNA or any nucleic acid. It's not just DNA, it can be any nucleic acid. So they are not the one that separates the two DNA strands. That is something different from cutting. Cutting, I mean, I'm cutting, for example, I had one long strand of DNA, I'm cutting this portion. This is done by nucleases. Okay, separating the strands as in I have this strand, now I am separating into two. That is something different. Now, exonucleases makes cut at specific position within the DNA. So nucleases, as I said, the enzymes which cut the nucleic acid. It can be DNA, it can be RNA. They are of two types. One is endonucleases and the other one is exonucleases. So endonucleases, if you see, for example, this is the DNA. I'll make it properly, and I want to cut it over here in the middle region. Okay, so this is done by endonucleases. On the other hand, if I want to cut it over here at the tips, okay, at the tips, if I want to do the cutting, okay, over here, who helps, children? It is exonucleases that helps. So here it is written makes cut a specific position within the DNA. That will be entirely wrong, my dear children. Okay, so what could be correct? Ligases. It joins the two DNA molecules. So you saw you had a plasmid, okay, and you put our DNA inside the plasmid and you wanted to ligate them. So for that you had DNA ligase. Okay, so this becomes the correct answer. Cool. Chalo, next question. This came in 2020. Okay, which of the following is related to genetic engineering? Heterosis, mutation, plastid, plasmid. So the answer is plasmid. Okay, what is plasmid? It is the extra chromosomal DNA. Okay, what is extra chromosomal DNA? For example, I have an Escherichia coli over here. It has its own circular DNA. So prokaryotes, no, E. coli is a bacteria, and prokaryotes basically they have circular DNA like this. They do not have linear DNA like us. So this is their actual genetic material. Apart from that, they also have other extra chromosomal DNA as well. Okay, and this is known as plasmid, my dear children. This is known as plasmid. Now, what do I want to do? In genetic engineering, basically I take my genes, okay, because my genes over here, suppose this is my DNA and I have very specific genes over here that is producing insulin. Now, I want to make multiple copies of this particular gene which is producing insulin because my body is not able to do so. Okay, remember this gene is for insulin. So, what do? I cut this DNA with the help of endonucleases. Okay, I cut it and then I put this in the plasmid. So I take the plasmid out, in the plasmid also I make the cut. Okay, now I have removed this particular portion and instead of this portion I have put my DNA, that is this particular DNA, and then I put it again inside. Okay, then I put it again inside. Now it will have like this, it will replicate. Okay, why I am taking a plasmid? Because over here, this chromosomal DNA which is there, it is coding for proteins and everything. This DNA which is there, it is not coding for any protein. So if I take, there is no problem occurring in the E. coli. Now, I am taking back the plasmid inside. This will obviously replicate. It will replicate. It will increase the number of copies, and that is what I want. I want to increase its number of copies. I want to increase the number of copies of my gene which I have put, this gene of interest which I have put, and now this will express and give me insulin later on. That is what I wanted. So here plasmid is of use, my dear children. Okay, please remember that in genetic engineering you definitely need plasmid. This came in 1999. Cool. Next question. Manipulation of DNA in genetic engineering became possible due to the discovery of what? Manipulation of DNA, as in any changes in the DNA strand. Any changes in the DNA strand, okay, that is what you mean by manipulation, came because of discovery of what, children? Because of discovery of restriction endonucleases. These are enzymes. So you had restriction endonucleases, okay, which were there helpful for cutting this particular portion of DNA and inserting it somewhere else. So I am manipulating. Now this DNA strand is not like this. This won't be having this big strand. Over here I have manipulated, and now there will be a small strand. You get it? So this is done by restriction endonucleases. When the discovery of this was happening, everything got better. Okay, genetic engineering got better, and you call them as molecular. You call them as molecular scissors, my dear children. Please remember, it's a very important question. What are the molecular scissors? These are the molecular scissors that you have. Works? This came in NEET 2002. Okay. Next question. 24. The cutting of DNA at specific location became possible with the discovery of what? Again the same question, 2015. You see the number of times the question has been repeated. So the selectable markers is the answer, ligases is the answer, restriction enzymes is the answer, probes are the answer. It is restriction enzymes. This was the reason why cutting of DNA was made easy. Okay, you know, cutting of DNA, for that we need certain things, and that certain thing is what? Just an enzyme which is known as restriction enzymes. Restriction endonucleases, they cut from within. Restriction exonucleases, they cut from outside. Okay. Next question. The eukaryotic genome differs from the prokaryotic genome because the DNA is complexed with histone in prokaryotes. Is it true? The eukaryotic genome differs from prokaryotic genome because the DNA is complex with histone and prokaryotes. No, children, they do not have much amount of histone there. Okay. Going ahead, the DNA is circular and single stranded in prokaryotes. Is it true? Yes, it is true. I told you right now, in prokaryotes they do not have linear DNA like us. They do not have linear DNA like us. Instead, they have circular DNA, my dear children. And it is written and single stranded in prokaryotes. It can be single or can be double stranded as well. Okay, that is entirely different. Now, repetitive sequences are present in eukaryotes. Is that the thing? Is because repetitive sequences are present in eukaryotes, children. Next thing, genes in the former case are organized into operons. In the former, as in eukaryotes, they are organized into operons. Both of them, they have operons. Both of them, children, they will be having operons. Okay, cool, works. Okay, just give me a second. I don't want to have a confusion regarding this. I don't know, my brain has frozen. Yes, so I don't know why even I had a doubt, but still. Okay, so this is the correct answer. Okay, as said earlier. Fine. So I hope you know that DNA in prokaryotes do not have histone proteins. Both of them will be having operons, so that does not make them different. Repetitive sequences are present in eukaryotes, so what? Okay, does not have a big deal. But over here DNA is circular, and that is totally different from us eukaryotes. We have a linear DNA, they have a circular DNA. Okay, going ahead, we have the next thing. According to Mendelism, which character shows dominance? According to Mendelism, which character shows dominance? Here axial is dominant. Here axial is dominant. Here yellow color is dominant, as I said earlier. Here round is dominant. This is dominant, and yellow is recessive. So this is the correct color. So the green thing that we choose is actually correct. Please do not get confused. Okay, so the green pot that we choose, it is the correct thing. The thing that I was getting confused is that basically inside the green pots you have the green seeds. You have the green seeds, right? So basically the seed coat should be green in color, the dominant one, but that is not the case. The dominant one is actually yellow. Inside this, actually it should be yellow in color. That's when we say, why is not showing up? It should be yellow in color. That's when we say that it is dominant. So yellow is the dominant trait in the case of the seed coat color, but in the case of green pod color, it's the same. That is why we choose it. I'm so sorry, but I hope I have rectified the mistake. Okay, works, cool. Chalo. In hybridization, capital T small t and small t gives rise to the progeny ratio. What? Let's see the progeny ratio. So this is the parent, this is the parent. You have to make the gametes. So for making gametes, this is on one chromosome, this is on one chromosome. Either this chromosome can go or this chromosome can go. So I am writing either the gamete can have capital T or the gamete can have small t. These are what, children? These are the gametes. Okay. Going ahead, again over here, it can be small t or it can be another small t that you have over here. Okay. Now, this gamete from this parent can fuse with this gamete, right? So in this case, it can be capital T, small t. This gamete can fuse with that, so it can be again capital T, small t. Over here, this and this can fuse, so it will be small t, small t. Over here, this and this will fuse, again small t, small t. Okay, so what is the ratio, children? Can you tell me the ratio? If you see the genotypic ratio, genotypically if they are same, so you see two of them have capital T, small t, this is 2 in number, then two of them have small t, small t. This again will be 2 in number. So the ratio, genotypic ratio is what? Genotypic ratio, if you see, it is 2:2. That is basically 1:1. This is the genotypic ratio. Now what about the phenotypic ratio? How do they look? Basically there is capital T or small t, there is capital T, small t. So capital T is kind of dominant over small t. Again capital T is dominant over small t. Okay, this would be tall, this would again be tall. Over here both of them will be, there is no one to dominate over this. Exactly, right? There is no one to dominate over this also. So this has to be dwarf, my children. So both of them are tall, both of them are dwarf. So what do I get? Phenotypic. If I write the phenotypic ratio, I got 2 tall and 2 dwarf. So 2:1:1. So the ratio will be 1:1 here and here. Genotypic and phenotypic ratio are matching. It is 1:1. So this becomes the answer. Cool? Works? Chalo, next question. Number of Barr bodies in XXX female is? Okay, listen to me carefully. What happens is that usually females have XX chromosome. Okay, 1X chromosome is the functional chromosome. And you know where are the chromosomes found? Basically, if this is a cell, inside the cell you have the nucleus, right? In the nucleus you have the chromosome. I say one is functional, the other one is not. The other one basically forms something like this, which is known as a Barr body. This is known as a Barr body. So in a female who has just X and X chromosome, one becomes non-functional and you call it as a Barr body. It occurs like a dark spot under microscope. Now on the other hand, okay, if the condition of the female is XXXX, which is given over here, okay, so you have one functional S, you have 3 non-functional among them. 3 of them, they would be what, children? They would be non-functional, and these 3 will be known as 3 Barr bodies. So your correct answer becomes 3. Now with this question I want you to solve one more question. That is basically, if the situation has now been twisted and it is asked to you that if this is the case, that is a male, what is the number of Barr body? Here you just have one X chromosome. Do you think that can become non-functional? No, children, it will be functional. So over here, if you are asked about the Barr body, there is no Barr body. Now where does Barr body play a major role? You might have studied about amniocentesis, right? What do you mean by amniocentesis? Basically, what happens is that for sex determination, people used to do amniocentesis. Actually, it is not for sex determination. It is for identifying any sort of genetic disorders. So what happens in amniocentesis is that, you know, in the womb, the baby is lying around in a space which is filled with amniotic fluid. So there is a fluid filled space, and this fluid is known as amniotic fluid, my dear children. So what happens is that this baby sheds some cells, okay, over here. So you have cells over here, and cells, if you see, they have the nucleus. Nucleus will be having the chromosomes and all the things. So in amniocentesis, there is a syringe being put over here. This fluid is collected. And then the chromosomes and everything, the genetic thing is being checked. So when they check it, if they find that basically there is a Barr body in the nucleus, that means this particular child is a female child. This particular child is a female child. On the other hand, same thing happens, but over here you are not checking for the, you didn't find the Barr body. That means it will be what? XY, and XY stands for what, children? Male. So this particular child will be male. So amniocentesis was actually used to find the genetic disorders, but since it was illegally used for finding the sex of the child, and then the female feticide increased, which is why it got banned. So you should be knowing in amniocentesis, it's the Barr body count that is being taken for the sex determination of the child. Works? This is the 28th question. Going ahead with 29th question, match the terms in column 1 with their description in column 2 and choose the correct option. Okay. So there is dominance, there is codominance, there is pleiotropy and polygenic. Dominance means, I will just directly tell you, over here if you have two alleles, capital T, small t, I will say this organism is tall. Even though it has an allele for tallness and an allele for dwarfness, why is tallness being shown? Because this particular allele is dominant over this. This is known as dominance. So this type of thing that you see over here, when it is capital T, both of them are same, you call this homozygous condition. You call this condition as homozygous. When it is small t, still it's same type of alleles, so still you call them homozygous. But when you have different type of alleles, you call this heterozygous. So the definition should be, in a heterozygous organism, only one allele expresses itself. Over here only one allele is expressing itself, and this is a heterozygous. So this becomes the definition for dominance. Okay. Now codominance basically means, for example, you have blood groups. Not everyone shows codominance. Not all the genes they show codominance. Okay, children. So basically you have blood groups A and B. If A allele is there, B allele is there, both of them will show up the character, which is why we have AB blood group. On the other hand, you have A allele and O allele. A is dominant over O. This is also heterozygous. This is dominant over O, and A will be shown just like this. Okay. Now, if B is there with O, again B will be shown because B is also dominant over O. But when both of them come together, A and B, they are codominant. So in a heterozygous organism, when both the alleles are expressing themselves, okay, it is known as codominance. So in a heterozygous organism, both the alleles express themselves. It is fully known as, it is known as codominance. Okay. Now what do you mean by polygenic features? So for example, you have skin color. Skin color is basically not denoted by a single gene. There are so many genes that is coming into action. So you have A gene, you have B gene, you have C gene. All of them, they show their effect for a phenotypic character, that is skin color. So there are so many genes involved over here. So when many genes governs a single character, that is skin color, you call this as polygeny. Okay, you call this as polygeny. Fine, works. Now there is something that is known as pleiotropy. Let's see what is that. Pleiotropy is when one single gene, okay, I am saying different genes are there, they are controlling one character. Now I am saying there is one single gene, that is A, suppose. This A gene has to control one character, agreed? Usually it is responsible for controlling one particular character, character 1. But A gene is actually controlling two characters over here. This is known as pleiotropy. This is known as pleiotropy. So there is an enzyme needed, okay, which is not present in the case of phenylketonuria because the gene is affected. Okay, so this gene is responsible for formation of one enzyme. But when this particular gene is getting mutated and it is not forming the enzyme, it is also responsible for other underpigmentation of, for formation of less amount of melanin also. So this means it is basically responsible for two characters, and this is an example of pleiotropy, my dear children. Okay, so pleiotropy, I can say a single gene inheritance influence many character. So this is the thing. So A will go with 2, A goes with 2, okay. B goes with 3, okay, B with 3, so here makes it all difference, okay. Now if you talk about C, C will be going with 4, okay, and D will be going with 1. This becomes the correct answer. This is the 29th question that we have over here. Cool. Now the next question that we have, hemophilic man marries a normal woman, their offsprings will be what? All hemophilic, all boys hemophilic, all girls hemophilic, all normal. Please try to solve it. I'm just coming in a second, okay. Yes, so hemophilic man marries a normal woman, their offspring will be what? Hemophilic man, as in, so man has XY chromosome, agree? Now whenever a man is showing the character of having hemophilia, that means the X is affected, because this is a X-linked recessive disorder. Hemophilia, please remember, it is an X-linked linked recessive disorder. That means if there is X and X condition like this where both of them affected, then only it will be shown. If the condition is like this where one is not affected, other is, then probably this will overshadow the effect and this person will be just a carrier. This will be a female and it will be a carrier. But in the case of them, do they have anyone to overshadow the X over here? They just have one X. So this is XC and Y. XC and Y. Okay. So over here, this is a hemophilic man and he is marrying a normal woman. He is marrying a normal woman. So normal woman can be like this or it can be like this also. It can be like this or it can be like this also, because this is also carrier, this is also normal, she is not hemophilic. Right. Now if you try to fuse them, so this and this goes, you have X, CX. Okay. Basically I can write like, okay. Now this and this goes, you have again X, XC. Okay. Now you have this and this, then it is XY. You have this and this, then that is XY again. So there are two males who are not having anything and there are two females who are the carriers. There are carriers. So all hemophilic, all boys hemophilic, all girls hemophilic, all normal, you do not have the option over here. So let's mark the next thing. Okay. So next condition is XCY and then you have that condition. Let's see now. So this and this fuses, you have XXC. This and this fuses, you have XC, XC again. You have this and this, then it is XY. You have this and this, then it is XCY. So over here if you see, there is one thing. So this becomes, she become a carrier. This is a female who is a carrier. This is a female who is hemophilic. This is a male who is normal. This is a male who is hemophilic. So all are hemophilic, children. Do you think all are hemophilic? No. Over here you do not see that all of them basically are hemophilic. They are carriers. So this becomes wrong. Can all boys be hemophilic over here? Okay. Do you think that all boys are hemophilic? This is this and this is the case. No, that is also wrong. All girls are hemophilic. All girls are hemophilic. No, children. One is hemophilic. So that is also wrong. All are normal. That is also not something that you can see. But over here if you see, this is a carrier, this is a carrier. They are normal. They are completely normal. So we'll go with this option again. Okay, they are just carrier. They are not hemophilic. They are just carriers. So I'll go with this and I'll say that all are normal and this becomes correct. Okay, now why did we do this? Because sometimes they just say normal woman, they won't say carrier woman. Okay, because they are also normal, they don't show this particular effect. Okay, they are just carriers, they are not hemophilic. If you are a carrier, you call them normal only, which is why I made both these things. Okay, now you can get your answer like this also. Okay, so that's all, children. I hope that you were able to solve this particular thing. So wishing you all the best and do let me know if I was helpful for you, and please practice mock tests regularly. It is very much important. That's all. Bye bye. Take care.